Well, good afternoon, everybody. It's a pleasure to talk to you this afternoon. I'm going to talk about my passion in my research life, which has actually remained more or less unchanged for half a century, which shows either I'm very boring or it was very difficult, or maybe both. I'm going to talk about how cells, and all living things, as you know here, are made of cells. How do cells control their division from 1 to 2? This is a very important process because it underpins the growth and development of all living things. When it goes wrong, you can get diseases, particularly cancer. But my interest has been driven mainly by it being a fascinating basic problem. It really, given that life evolved from a single cell, it's really the primordial developmental pathway, developmental sequence. It underpins, thinking underpins thinking about temporal order, how do events occur in a particular sequence, what is the pacemaker that drives the overall progression through those particular events. Now, cells reproduce through this process of the cell cycle, and you see here a mammalian cell just completing its cell division. And there are a sequence of events that have to occur for this to be a successful event. And this characterizes it, and this is a figure I drew, I think, 40 years ago, and I still use it to remind us that we have hands, fingers, and pens, and not just keyboards. And what you see here are the two primary events of all cell cycles, which are S phase, when chromosomes, the hereditary material, are copied. Blue goes from one chromosome to two chromatids there. That's S phase, the replication of DNA, and mitosis, when those two replicated chromosomes are segregated into the two newly divided cells. That gives you mitosis and cell division. These are not the only events that occur in the cell cycle. They are the primary universal events because they are essential for each newly divided cell to receive a full complement of genes. And it's this process, common of course in all eukaryotic cells. Prokaryotic cells have the same problem to solve, but do it in somewhat different ways. It's this process that I've spent my life working on. And particularly at the beginning of this, there were several others, and I'd like to acknowledge Tim Hunt. Where are you, Tim? Are you somewhere in the audience? I'm sure. Late from lunch, perhaps. Late from lunch. Tim Hunt, my friend and colleague, and Lee Hartwell, who's not here. We were three who were working on this problem very early in the 1970s. I took, following actually the leadership from Lee Hartwell, who worked in budding yeast, and I was only a graduate student when I first read his papers, I took a genetic approach to this problem, working with the fission yeast Schizosaccharomyces pombe. And a genetic approach means you look for mutants that are altered in the process that you're interested in. And my starting point, and you see up here was the first two papers I published in this area in the mid-1970s. And you'll see A there, that's a fission yeast cell. It's a cylinder, it grows by tip elongation and then divides. And I looked for temperature-sensitive mutants following what Lee had done in budding yeast, and simply looked, very simple, simply looked for temperature-sensitive mutants which at a high temperature, they have to be conditional because they're essential, which failed to complete a cell cycle progress, but could continue to grow. Because obviously if you block protein synthesis, RNA synthesis, you block cell division, but that's trivial. What you needed was something that was specific for the cell cycle. So I went through mutants, collected mutants, identifying CDC mutants, cell division cycle mutants, hoping to build up a picture of how the temporal order might actually work. Then I stumbled by mistake. I like to say that I thought it through and looked for it, but I didn't, completely by mistake, a mutant with the complete opposite phenotype. And you see that down there on the AB pair, a different sort of optics. And these were cells that were dividing at a small size. I found them in Scotland. I called them wee mutants. I thought it was funny at the time. 50 years later, the humour is a bit thin, but I still have to explain it, of course. And these mutants allowed us to do, if you like, a logical analysis of the cell cycle to address, let me remind you, the two problems. What is the basis of temporal order? What is the pacemaker of cell cycle progression? And the wee mutants were particularly important because they identified the pacemaker, because these cells divide faster than they can grow. And so they are rate limiting for the overall progression. As I said, I'd like to think that I thought of that, but I didn't. Nature presented it to me, although I did actually notice it. What was interesting was there were very few mutants that could give you that wee phenotype. And one of them could be mutated in two ways. Like the top one, which was very common, or fairly common, it couldn't divide, but particular dominant mutants could be advanced through the cell cycle. This encoded a gene called CDC2, which was briefly mentioned, and that turned out to encode what we now know as cyclin-dependent kinase. Tim Hunt, if he's returned from lunch, but I don't see him yet, took a biochemical approach here and defined one of the two components, because this enzyme is traditionally thought, and I'm going to briefly mention an advance at the end of my talk, only of two components: a protein kinase, CDK1 often called, which is encoded by CDC2, and cyclin, an activating component of the kinase. Cyclin was the protein that Tim said disappeared. There's very few proteins that disappear through the cell cycle. We now know that from proteomic studies that we've carried out. But this is the protein that disappears, and disappears at the end of the cycle and then reappears as cells proceed through the cycle. It's an activating component of the protein kinase. Why is the protein kinase important? It's a kinase is an enzyme that phosphorylates, puts a phosphate group on other proteins. And they can act as switches, because putting a phosphate group changes the activity of a protein, so you can regulate many different proteins. And in the case of CDK, we have now in the lab, using in vivo studies and mass spec, identified 400 CDK phosphosites on about 250 proteins. So it's a very extensive set of substrates that are phosphorylated for these processes. So the idea that it was developed around 2000, when the Nobel Prize was awarded, was that there was a family of different cyclin-dependent kinases, different CDKs, different cyclins, that these had different substrate specificities, which were required for the different events of the cycle. So this was thought to underpin temporal order. And the appearance and sometimes disappearance of these CDKs was brought about by different regulators. It got really quite complicated, a little bit sort of messy. And I now want, since 2000, and it sort of is really an example of how science evolves, is tell you about things that I think should now be looked at a little bit differently as a consequence of subsequent work. And the first thing was, in an attempt to simplify the system, hoping that it would reveal new information, my lab deleted all the cyclins, and it only has one CDK, and showed that we could get a perfectly normal cycle with only one cyclin-dependent kinase. Now that blows a hole in the model I just said. I mean, it shows you at least one eukaryote does not require all these multiple CDKs. Given that, we thought we should look at the substrate specificities in vivo of the different CDKs that had been implicated. There were 4, as I said, at different stages of the cycle. We did that in vivo, basically using shock-at mutants to kill the kinase, and then measure, by making fusion proteins with different cyclins, and then measuring in vivo the CDK activity. We just killed the activity. It's like doing an in vitro assay, only in vivo. We kill the kinase, everything gets dephosphorylated. We then remove the inhibitor, and then simply do a time course of rephosphorylation of all the substrates. And this showed that, for example, the G1S CDK versus G2M had actually 90% identity in terms of substrate specificities. So the idea that they have very different substrate specificities does not actually occur, at least in the fission yeast. So it is something in more complex organisms that may be involved, but it is not a basic principle that's going there. So one is we can run it with one CDK. Secondly, the substrate specificities are much the same. This led to this model of which I call a quantitative model, which is much simpler than the model you'll read in the textbooks. What we say the basic principle driving the eukaryotic cell cycle is simply a rise of total CDK activity in the cell, acting like a rheostat that phosphorylates a set of proteins at low level, which are required for S phase, and increasing levels as you proceed through the cycle till you reach mitosis when there's a burst at mitosis. Of the 250 substrates, the majority, over 200, are required only actually at mitosis. So this was different from the, as I said, the textbook model that you actually read. But if you think about it, in the primitive eukaryote, since CDKs must have arisen by gene duplication, this is probably what must have happened. Now, I said total CDK activity. So in the last year or two, we've shown that isn't quite true. It is the CDK activity in part of the cell, which is the pacemaker for the whole cell cycle. And that's shown here. We developed sensors, in vivo single cell sensors in the last couple of years that allowed us to measure CDK activity within the nucleus and the cytoplasm. And using different colored substrates, we can do it simultaneously. And what you can see here is that we've measured the activity here in red in the nucleus, which goes up when you get S phase around here, and then is activated, and then in the cytoplasm, there's a 10 to 15 minute delay, approximately. So what we suggested from this is that actually it's not total activity, it's the activity in the nucleus. The nucleus is only 8% of the cell. So if you just do in vitro extracts all the time, you will mix up the control systems, which turn out to be different. We also show, but I'm not going to show you the data, that the active CDK in the nucleus is then exported into the cytoplasm, and because of positive feedback controls, it activates the CDK in the cytoplasm. So cytoplasmic total activity is a slave on that in the nucleus. So it isn't total cellular activity, as we'd argued. It is actually nuclear activity that acts as the pacemaker. This is a bit technical for those who are into phase plots. This is an in vivo phase plot rather than an in vitro one, which is normal. And what this really shows for those who are into this is CDK activity. This is the level of CDK. And when you get a trace like this, it shows it's very strongly regulated. So in the nucleus, it's very strongly regulated. In the cytoplasm, it is not. So this is very prone to adding some active CDK because it's not tightly controlled. Also, remember the checkpoint controls that occur at mitosis have to make sure that S phase is completed. And where is the DNA? It's in the nucleus. So it all makes completely sense. In the last 37 seconds that I have, I'm going to give you my joke Friday experiment. I do experiments in the lab with my most junior technician, who I then train and they go elsewhere, and they do experiments that no proper postdoc would contemplate. This is my Jurassic Park experiment. Because of the conservation of CDKs, we can compare all sequences, predict what it looked like at the birth of eukaryotic life, synthesize it, and see if it still works in an organism like fission yeast. We've created it for CDK1 and for cyclin. We're making now a cell that is dependent upon, it's not the Jurassic Park, it's about 1.5 billion years older than the Jurassic, and actually it works. Not the cyclin, haven't done that, the CDK does, and a third subunit, CKS, which we also are working on, does do that. So thank you for listening to me. It's been very exciting to be doing this recent work, but the main message is science evolves. What we thought 20 years ago isn't what we thought now, even though it may have won a Nobel Prize. Thank you very much.